And then again, Michael's put a reminder here, MVZ retreat next week on Wednesday. So it's my pleasure to introduce Vladimir Pravosudov. I got to meet Vladimir with my connection with SageGen, where he does a lot of his chickadee work. But before that, I'd read a lot of his chickadee papers looking at this amazing system that he's developed between investigating cognition in these birds and how they store food resources. And it was only after meeting him at SageGen that I sort of clicked, well, wow, he actually did that work on one of our NRS reserves. So it's a real pleasure to have him here visiting today. He is going to be telling us about some of the work in that system, but some of the broader work that he's done as well. So thank you very much.
Thank you. Thank you very much for inviting me. So I did use a resource of UC Berkeley for a long time. I work at SageGen since '99, so that's been a pretty long time. And today I'm going to try not to burden with much data, but even though it's very exciting because of my students, we have data streaming in, so I'm also adding new slides all the time, and hopefully it's not going to be too much. So I always try to overcrowd my slides with kind of things. All right, so the overall kind of idea, I'll try to focus on mountain chickadees and bring it to the bigger context, what we've been studying. And again, the main idea for us is to understand the evolution of cognition. We're using a smaller system, a population type of approach system, where we're trying to find a natural model where we can see the evolutionary processes leading to the diversification, divergence in different cognitive traits. And so the basic idea, again, is to understand what are the driving variables that provide this difference in selection on these traits, what are the maintenance variables. In other words, we're trying to link it all the way from environment to cognition, to the brain, to physiology, and then back to the whole ecology, trying to understand if we're dealing with the system, populational processes, and everything else going on and controlling on it. And the mountain chickadee, the system, which is a gorgeous place. So again, you at Berkeley probably have been at SageGen, that UC owns it. It's absolutely spectacular location. I'm lucky to live 20 minutes away from there, so it's literally just like getting out of my porch, and then 20 minutes I'm in the field, and I love it, so we go there all the time. But before, again, the way that this research started was not necessarily from the SageGen, and even though I did some basic work on this chickadees, just trying to understand general processes controlling memory. So yes, we are focusing on these food caching animals, which are pretty spectacular animals. And again, the underlying idea of this, so this is not my picture, but I love this picture because it reflects the essence of the chickadee, so to speak. And it's a black-capped chickadee, that's how it started. And I've done my master's for a long time in ions before in Russia using these birds. And I was always fascinated when I first started working in biology, when I was still in high school, kind of finishing it. When I would go to north above Polar Circle, I would see these birds, cheerfully, moving around when you have 2 hours of light and minus 40 degrees Celsius, and they're doing just fine. And that kind of initially started, how can they do that? I mean, people cannot do this, we cannot survive. These birds have, 3 hours maybe at most to forage in twilight, and then they sleep all the time. It's a 12-gram bird, how they do it, right? And so an initial idea then led to understanding that they actually cache food. So I kind of followed the work by Svein Haftorn in Norway, who was a pioneer of this work, and then I realized that that's a big system for them. So they store a lot of food in the fall when it's abundant, and then they use these caches to survive the winter. So my early work showed that individual bird, like black-capped chickadee or willow-tit, can cache up to 500,000 individual food caches in 1 year. So that's kind of mind-boggling. 1 million food items, right? So that's a lot. That's all they do in the fall. They breed, then they cache, then they retrieve, then they breed, then they cache, then they retrieve, and then they either die. No, that's a whole story saga of these birds. Right, and then it led me to kind of think about natural system of these things, just looking how much they do, what's happening, and then I was interested in the mechanism, so that took me to NIH and kind of getting into the brain, and that's what I'm kind of using the system where you have the entire connection. And so the underlying idea again is that natural selection acts on it, right? And so we have this main premise is that food caching is equal fitness. In other words, if animals don't cache enough, they wouldn't be able to survive, right? With this, you can come with multiple predictions that when it's more difficult for them, they should cache more. And we'll talk more about this, and they all confirmed that they do indeed cache a lot more when winters are more severe and conditions are more difficult. Cache retrieval is dependent on spatial memory. That's unquestionable now. I'm not going to go into a lot of work that multiple people, including me, did connecting this, that if you impair spatial memory in any way, form, or shape, you will not be as good at retrieving caches. So spatial memory is dependent on the hippocampus. Obviously, there are other parts of the brain that also are involved, but hippocampus is a gateway of memory, and so we focus on that, right? And then we can have this reliance of harsh environment. It basically provides more dependence on cached food, and that relies on more memory. So in other words, if you have harsh environment, basically that should select for better memory in the birds. That allows them to catch more food and to successfully retrieve it, right? And again, the idea with this would be that if you're not good at it, you're probably not going to survive. So we're talking about very high mortality in the systems. Probably 40–50% of the population goes out every year. So in a sense, it's like a little Drosophila model where you have a very big sweep every year coming through population, taking away some individuals. The question is, who is going away? Are we talking about the ones with bad memory, and so we have direct selection on better memory, or there are some other processes? And also, when you go into mild environment, I always have questions saying, why are they so bad at memory? And they're not bad, they're just not as good. And then the question is, why not good, right? And so we were talking about studying some mechanism that prevents the distribution. So if selection acts on and being smart, then these birds are not just, can't come back around. And we'll talk about other things that may prevent them, because it seems from our studies and other studies that evolution of memory leads to lots of other changes in their behavior, and particularly as well focus on mountain chickadee system. Smart chickadees, for example, are social subordinate, less aggressive. So in encountering with the low elevation chickadees, they always lose. And so in that way, yes, they're smart, but they can't quite penetrate because they're not going to be successful. And their superb cognitive abilities are not going to be any good for them because they don't need them there. But at the same time, they're going to get their behind kicked every time they're going to try to contest for a female or something like that. Right? Again, this is just a background. An initial thing, again, I'm going to throw kind of a bunch of data. I'm going to try to make sure that it's explained. So this study first led me into trying to think about testing it in a system that spans over a large range, geographic range, right? And that is a black-capped chickadee. So here's a range of a black-capped chickadee. As you can see, the main thing when I first saw it, struck me is that if you look at the north-south type of the distribution, that it's a gigantic difference in environment, right? Specifically the winter. So if you go here in Fairbanks, they go as far as the forest range goes in Alaska, and so up there we're talking about pretty severe winters compared to anywhere down there, and so you can basically generate this type of gradient. So we've collected birds along this gradient, and you can see we'll look at a lot of things. I'll just show you hippocampus, and we'll look at the hippocampal volume and the number of neurons in the hippocampus, and they show a really nice relationship across these ranges, right? And again, we're using this more not like a direct regression by a specific degree of change, but we're seeing there's a relationship, right? Basically, the far north you go, the bigger hippocampus you have, and that's relative to the brain size. It means they just have bigger hippocampus. And of course, we also show they have better memory, they catch more food under controlled conditions, and so on and so forth, right? So that nicely supports this idea on this. We also wanted to see, are we talking about temperature and snow, or are we talking about day length? Like I mentioned before, usually the first thing that strikes somebody about being in the north is a very short day in the winter. And I work in the field there, and when you only have 3 hours a day that you can be in the field, and then you're stuck for 21 hours in the cabin with a couple of your colleagues, that is a tough condition. Because you can't go out because it's dark. It's pitch dark and you're somewhere, right? So, and so we want to see if that's a driving force, because clearly daylight limits foraging opportunities, so they cannot forage. So we wanted to do the experiment where we would compare, because the temperature and winter severity also ranges latitudinally, right? So we can compare Maine, for example, Minnesota, which is pretty tough as well, actually colder sometimes in Alaska, and Seattle. Seattle is very mild, right? So the question is, are we going to see the same differences? And we do. So we actually see the birds in Maine and Minnesota have the hippocampi size the same as in Alaska, and they have the same or similar number of neurons, while birds in Washington and around Seattle, they are more similar to birds from Kansas, from Iowa in this case, with a milder climate. So which basically tells us that we cannot rule out that the day length is important, but just the winter itself seems to be associated with these differences, right? And when we think about, again, the winter, we think about snow, we think about cold. These birds normally are insectivorous, so once the winter comes, there's no natural food for them available. So basically they are relying on their caches. And the longer the snow stays, the less ability they have to get their natural food. And so they need to rely more on caches. So initially when we did this, we're looking at the big scale, and I probably don't have to work hard to convince you that Alaska is more harsh than Seattle or even Kansas. But then I kind of thought, because I've been also working at Station, and I thought, what about the mountains? Yes, it's not as tough, and people may think, well, how tough could it be? We're in California, and it's beautiful, Lake Tahoe, it's not bad. But when you think about it, if you go to the top, if you go to 8,000 feet, and then you go down to 6,000 feet, you probably, if you think about it, you will see that there's a very predictable and reliable pattern. The snow level, every time you listen to forecasts, it says rain below 7,000 feet, snow above 7,000 feet. The snow comes 2 months earlier, it stays 2 months. Right now, there's terrible snow. We still have full snow coverage at above 7,000 feet at the field site of Station, right? And so the question then is, these birds are occurring over the whole range, so can we see the same type of a pattern of a tiny spatial scale? Because unlike the continental scale, when you go up, you go fast. I mean, you go just 10 miles, and you go from 6,500 feet to 8,000 feet. That's very quick, right? So, and initially when I started doing this, I just got a new master's student, and I thought, why not do it? And I had all my postdocs saying, don't do it. There's no way they can see something. It's too close. It cannot happen. You're going to waste all the resources. And I said, well, it's a master's student. Why not do it? I mean, we can teach a project. We can do this. It's a cool idea. And so here it is. I took this naive master's student who came to me, and I said, let's just do it, right? And so initially, again, we just sampled 3 elevations. So we went there, and we collected birds and brought them to the lab. And what do you know? We see the same pattern, right? So if you look here, this is a memory test and one trial associative learning task. So the one trial task means that the animal goes in the room, you provide a food where you want it to be, and then the animal finds it, then you remove the animal out of the room, and then after a retention interval, you put it back in, except now all the locations are closed, so they cannot just see, they have to find it. And then we count how many sites it will open in order to find the correct one. So the smaller the number, the better. So basically this is a much better performance than that, right? So it means this one that they found an average on every second try. And we usually give them like caching locations, so we do like 100 locations in a room, right? And so when you do this, the birds from high elevations are doing much better, right? And so we can look at number of caches. They actually cache much more, which is also interesting because that's in the lab, and the initial data come even before we start quantifying it. We had undergraduate students cleaning cages, and we had these birds, high elevation, low elevation, high elevation, low elevation. And my students came and said, how come every other cage has a lot of seeds under it every time we clean it? And the way they cache in the cage when you can't let them do it, they just throw it out of the cage. They think that that's safe. And so what you have is a bird from high elevation stores a lot of stuff outside the cage, so you have to sweep it every day, and the other ones don't do it as much. I was like, ha, we're onto something. And when we test them in their room, sure enough, we got the same pattern, right? Even though they're in a nice environment, they're constant for the period, everything else, yet they have this higher caching propensity. Then we tested them on a regular associative learning task, which means now the food is always in the same location. So the animal goes in there day after day, and it's always there. So you assume they will learn, right? So because you come there and you know that food is in this drawer. And you can see that initially there's a difference. High elevation birds were very good at the beginning, and it took 4 trials for low elevation birds to converge. They did converge, they learned it. It just took them longer to learn it. And then once they learned it, we gave them 2 more trials. Then we separated by 2 weeks, just to see the retention of these memories. And as you can see, the high elevation birds, they remembered really well. The low elevation birds forgot more. They were not bad. I mean, again, 6 out of 80 or 100 sites is very good, but it's just not as good, right? So you can clearly see the difference. So now we know that the difference is both in accuracy and in retention of memory, which is kind of very interesting, right? So they can better identify location, but they can remember it also for a longer time period. And these are the birds that are separated by just 10 miles across the habitat, right? So we'll have these 3 elevations. So we looked at hippocampal volume. We see a nice relationship the same way, right? If you're low elevation, you have smaller hippocampus. We'll look at total number of neurons. It's not linear, but this is still significant. There's a big break here, which is interesting because that is a bird at 6,000 feet. That is at 8,000 feet. The biggest change in environment occurs at about 65, 7,000 feet. While if you go lower, it doesn't change drastically. In other words, it's not linear change in the mountain conditions. It goes bad sharply, and then it starts going slower, right? So, but we still the same pattern, right? So we look at the number of DCX cells. We're looking at neurogenesis. So I'm not going to go too much about neurogenesis, adult neurogenesis. You may have heard about this. It's an important process, but we now know that new cells are being constantly produced in the hippocampus. Birds and mammals and reptiles and most vertebrates, and they've been implicating an important memory role. So the way we test this, we basically use a marker that identifies juvenile neurons before they mature, before they are 6 weeks of age, which means they're newly born, what we call DCX. And what we can see is that these birds are producing more new cells in the hippocampus, quite a bit. If anything, actually in birds at high elevation, chickadees, they have probably on average about 30% of all the cells that are new within 30 days of age. That's a massive amount, which means we have a lot of exchange of the cells going on specifically at high elevation. And so here we're looking at the soma size. You can look at this picture, you probably can see it. You can see how big the cells are in the high elevation chickadees compared to this one. So you can see the soma size here, right? This is also just a picture of the density. You can literally see this with the naked eye. I've always been very skeptical about doing statistics by eye. These are big differences. I mean, when I saw them, I was like, whoa, and somebody said, oh, you probably cherry-picked the area where you took the pictures. No, we didn't. We just put them in the same location and took the pictures. The difference are really that big, which is amazing, right? So they cache more food, they have better spatial memory, both accuracy and retention, they have larger hippocampus, more and larger neurons. We've done more neural work, all leading that there are real differences. They're there on small scales, right? So the last work, well, it's not time, in the time-wise. We also wanted to see are they good at problem solving. So at least in comparative psychology, evolutionary psychology, that's a big thing. You know now whether animals can do better at solving novel things, right? Sometimes we can struggle. What does it mean ecologically? You know, how irrelevant it is. So when we think about animals invading cities, that may be relevant when animals encounter new environment. But we can also think that animals encountering new environmental conditions, new puzzles that they have, can they do better? So we use this pretty simple paradigm with this. You can see that there's this vial that has a mealworm in it. They like the mealworm, and it's simply plugged with this cotton. So the bird has to go plug this. The mealworm will drop, and then they can basically get this. So let's see if we can play it quickly. Yep. You solved it. Obviously, you know, it wasn't that easy for them the first time, but for video, we always want to use this. Like when BBC came to film it, they wanted just to see the fastest, the best way. And then it took us hours and hours of training and doing this, and then they showed like, oh, here it is. And, right? But anyway, high elevation birds indeed took fewer trials to solve these problems. So we see a similar pattern. We also show this in black cats as well. So we see the similarities. The question with problem solving frequently comes to, is it cognition or is it something else? Because if animals, for example, more neophobic, then they may not be good at it because they may not approach the novel problem as easy, and so we may not be measuring what we think we're measuring. So we'll look at neophobia, right? So we did two different tests, and one of them, we basically train the bird to come to the regular feeder, and then once they train, they learn to go there, and then we replace it with a scary, red-looking, you know, transformer type of feeder with all these other things, and we try to increase the intensity. We put this big chickadee cracker box on top of them, and so we try to progressively make it scarier for them to go to the feeder, and then we measured latency to perch on the feeder to take food. And so you can see that our progression of scariness did work. They actually were more and more neophobic, but there's no difference between high and low elevation birds. They showed the same degree of neophobia in response to that. We wanted to replicate at some point and do it in cages. So the other test was done in an experimental room where the bird would fly in when we do the memory test. We also wanted to do this in a lab or in a cage where birds may be more comfortable because it's their home cage. They spend their entire life there. They do a lot of things. And then we randomly assigned one of these also scary-looking feeders to them instead of a normal one. And then we looked basically how long it takes them before, so we can see this pre-touch, pre-land, pre-take, and that's a regular feeder. As you can see, it takes them no time. They go off to the waxworm, which they really like. They like them a lot. And then we have these feeders, and again we use the same, the pre-take, you know, neo-touch, neophobia touch, and neo-land. Again, they're clearly neophobic, but there's no difference between elevations, right? But what's interesting, when we compared the individual performance on problem solving and neophobia, we actually showed that more neophobic individuals were better at problem solving, which actually goes exactly opposite to what sometimes people think. I can't explain this result. It's kind of interesting. We actually didn't think about that one until the reviewer suggested, did you look at it? And we said, like, oh, we actually didn't. Let's look at it. We looked at it, and yes, it's in the paper. It's kind of really interesting. If anything, it strengthened our conclusions that difference in neophobia, in problem solving, are kind of real, and they're not connected to neophobia. If anything, birds that are more skimpish, more kind of scared of new things, are actually better at solving this novel problem. So the question then, of course, is what's the difference, right? Frequently when we think about cognition in the brain, a lot of emphasis is always placed on plasticity, right? So when you read all this in a lot of psychology, all this brain is plastic, it gets bigger, we are learning, we're doing all these things. So is it possible that what we see here is just simply plasticity? So the animals are being placed in the harsh environment when they're forced to do this type of things. They just get better, right? Versus when really we're talking about selection on some traits that could be highly heritable. And I would like to say from the beginning, we don't have data on heritability. That's one of our ongoing work. But we did look at it in humans. There's actually pretty good literature. And I always like to tell my students, actually, a lot of our cognitive traits are highly heritable. Our spatial memory, our verbal abilities, our IQ things heritability levels are above 40%, which is pretty high. So when I teach my class, I always tell my students that if you don't do well, blame your parents. So, but it's interesting, right? So I think that we're trying to get pedigrees on these things, but the point is, it's very important for us to understand, because if we're dealing with very plastic responses, we can better predict how animals will deal with changing environment, whether they can simply respond to these changes, because normally plasticity is the one to prevent or slow down the evolutionary process, versus when we have a lot of variation associated with genetic variation, and through simple selection, or not so simple, we may see changes in allele frequencies in these populations, even on a small spatial scale, right? So in other words, we can be dealing with local adaptation and natural selection. And we want to know what's the extent of contributions of these two mechanisms. So one work we did with black caps initially, because that was a key question, right? So we basically, we did a common garden experiment, kind of old-fashioned type of experiment where we went to the nest boxes, we collected these birds before they even opened their eyes. So they didn't see anything, and we brought them to the lab, and they, all their life they spent in cages, right? So if there's any differences still there, that couldn't really be because some environmental things were affecting them when they were caching or not. We were not surprised, I guess I was not surprised, to find that the difference was still there. They were pretty strong. So the birds from Alaska were still, they were caching with food. These birds spend their entire life in a cage. They've never seen unpredictable food. All the food was amazing, and yet they were still caching more, and their memory was better at the same thing. The most striking results, which we've been kind of seeing again and again, the number of neurons were not affected at all. So if you can see here, this is the number of neurons of the lab-reared birds, and this is the number of neurons of wild birds. So in other words, the bird that spent its entire life in a cage about this size has the same number of neurons as the bird that roams the wilderness of Alaska its entire life. So to me, it's as drastic a difference in environmental enrichment as one can only imagine, right? So it was kind of very striking and interesting, suggesting to us that there's some developmental mechanism that controls the number of neurons that may be under pretty strict genetic control, and we're kind of still pursuing these things, right? And so in mountain chickadees, we didn't quite do the same thing, but we found all the differences in juvenile birds before the first winter. In other words, if you collect the birds at unknown age or adults, then we may be sampling the ones that survived, and so our results could simply indicate what's coming out after the selection event. So we actually looked at the birds before the first winter, and we saw these differences. So it's interesting, right? It's also supportive of the same idea. So significant differences also remain in birds that we brought into captivity and maintain in a uniform environment. So you can see the hippocampal volume. What I want you to kind of specifically address here or see here is that the hippocampus does shrink. So hippocampal volume, that a lot of people, or brain volume that people measure, does respond to environment. That's about 30% shrinkage. There's still significant difference between high and low elevations. But the hippocampal volume shrink. Number of neurons don't change. Which is kind of interesting, right? Suggesting what is the mechanism then? Because frequently we see a lot of evolutionary studies relying on volumetric measures. What do they mean? And so what we're seeing here, a lot of volumetric changes occur via changes in glial cells, changes in soma cells of neurons, which are well known to be changing very quickly in response to environment, and dendritic branching. And so when you think about it also, it may make sense purely from economical, if there's such a word in biology kind of perspective, where to generate a new neuron is very expensive and time consuming. If you have a neuron in place that's very stable, then to do everything else is very quick. So we know that animals can respond by changing dendritic branching within minutes of changes, right? So in other words, these things can occur very rapidly. But at the same time, the number of neurons don't seem to be affected by long time in captivity. We also have another striking result that I'm still we're trying to publish, but I'm not going to talk too much about it, right? So what does it mean that they shrunk the hippocampal volume? So we often think about this like clinical effects, like your brain shrinks, it's terrible. You don't have enough power. So in the end of these experiments, we release birds. We take them back. But we release them in spring, so they spend 6 months in the lab, and we take them back, but not quite to the stage we can't get there because of snow. So we release them about 15 miles from location. So when you release them, you bring high and low elevation birds in the same thing, in the same truck, we release them. All birds come in one tree, and they separate high elevation birds, the whole group, take a course at high elevation, boo, and they take off. We have 50% return rate to exactly the same locations within days. So they have 30% smaller hippocampus, they have no problem finding where they came from. They come back right there. And then they breed. Actually, one time we released them in mid-April. In 2 weeks, we found several birds already paired up, and with a female and a nest. So that's pretty impressive to me. Not only that the phylopatry is so strong, that they're coming back, but they're actually doing it with a hippocampus pre-shrunk by 40% or 30%. So that doesn't seem to affect their navigational abilities, right? So we also did other experiments trying to understand how plasticity or experience may feed into these differences. And so you can see here we brought birds in the lab, and then we deprived some birds from any caching experience or memory experience. We kept them in the cage, we gave them powdered food, we didn't allow them to store any food. And then we had another group, what we call experience group, that were allowed to cache and retrieve and engage memory on a regular basis throughout the entire experiment. So here's the hippocampal volume, here's number of neurons. But you can see a confirmation that the hippocampus does shrink. Again, that's a different experiment. So we clearly see this pattern. It's not a fluke, it's real. So the birds, you bring them to the lab, the hippocampus shrinks by 30 to 35%, right? Any changes in experience have no effect on the hippocampus at all after that in the lab. The number of neurons doesn't change at all, right? We have other things that we found. Neurogenesis rates actually being significantly affected by this, but not these other variables, right? So if you look here, again, the large number of environmental changes, but you see that, look how many more neurons here on the same scale, and see how bigger these things are. I mean, you can literally see that, right? But it looks like these changes in the number of neurons, I mean, they're just not happening. So in other words, you bring a bird, you put it in any kind of environment, the number of neurons don't change significantly. It stays pretty much the same. So that kind of suggests that, again, that we can be talking about pretty high heritability on that currency. There's also now there's a work in mammals suggesting the same thing, that number of neurons is a much better predictor of cognitive abilities than any other variable. So that our case kind of feeds into this. So what about development, right? So development is well known to affect cognition and phenotype in adulthood. So is it possible that birds at high low elevations are more stressed, and so as a result they have these differences, right? So, well, I mean, if it's likely, then we should see that birds at low elevation should be more stressed, which is not really likely because it's a more mild environment. But the bottom line is, we actually found no differences. So we looked at the development on both elevations, and they're developing exactly the same. So it doesn't seem to have any traction for us. It's not to say that we've seen everything. Obviously, we didn't measure a lot of things, but just basic things, and like things like fluctuating asymmetry that we specifically tried to look at. We didn't see anything. Why I'm bringing fluctuating asymmetry? Well, previous experimental work that I did a long time ago when I was still in Davis showed that if you stress even slightly nutritionally birds in a nest, just during very short time, and then you measure these things months and months later, there's a very strong differences. So in other words, the nestlings that suffer some shortage of food have much higher fluctuating asymmetry in multiple measures. We don't see any of this in our chickadees at all. So we don't know. We don't think that development is acting. Again, this is still on a more correlative, speculative nature, but we're trying to accumulate more and more. We're kind of leading and suggesting the same thing. So basically, what we're thinking is now we are thinking that the environment really kind of puts a borderline or an edge between these things where there are different processes controlling these population processes at high and low elevation. And if that's the case, we should see divergence in other behavioral traits and also some evolution of some isolation mechanisms, right? Whether it's when we're talking about mating, whether there's a female choice, song, and all of these other variables. Then the birds could see and say, yep, that's a guy from high elevation, I like him, or that's a guy from low elevation, I don't want to mate with this person, if we see these things, right? So we wanted to look at other behavioral traits that in the past had been always correlated with cognition, such as, for example, novel environment exploration rates. What do we mean by this? So usually the way of the testing, you take a bird and you stick it in a novel environment and you see how quickly it will explore the environment for the first time when it sees it in its life. So interpretation may vary what it means, but it's a good repeatable measure that's frequently used in personality studies. So we're not looking at personalities here, we're looking at more population level differences. But still, so we have this type of rodents and we let bird in and then we just see how many substrates it will explore over a given period of time. And so you can see here we counted 2 minutes and all the way up to 10 minutes cumulatively, and there's a big difference. So we can see that low elevation birds are much faster explorers, which technically goes consistent with the previous literature suggesting that smart animals are usually more thorough and slower explorers. Whether we're dealing with some kind of a syndrome or correlation, we don't know, but we do see similarities in this way. So these birds are smarter, but they're or not as smart in terms of their cognition, memory, and problem solving, but they're faster. So in other words, maybe they're just moving fast, exploring it fast, but not paying attention, right? So like quickly, and these guys are very thorough, they're doing this. We also look at just their activity patterns, like flights, because sometimes animals will fly and come back to the same substrate. That's not exploration. We also see pretty big differences, so that low elevation birds are also more active. They're not just exploring more, they're flying more. So clearly they're different. That's kind of also the bottom line, right? And so what does it mean? We don't know. We don't know whether selection, potential selection learning results in correlated, response of multiple traits, because one can also speculate and predict that these other traits could be also beneficial in harsh environment. And so in a way, selection can affect all of them independently at the same time. And we didn't intend to really try to understand them. What we also wanted to see as the next one, kind of going more into the natural history of these chickadees, we want to see if there's a potential trade-off between cognition. So if chickadees at high elevation are smart, is it just, so they're just smart and they're better at everything else? And the impetus for this study came from another study from Oxford, in which they actually showed that smarter gray tits were actually not as good parents. They were actually abandoning their nests more quickly. And they kind of didn't say it's a trade-off, but it kind of suggests that. And actually, if you look at human literature, there's definitely a lot of suggestions saying that high-level intelligence leads to fewer kids or no kids. And so there's suggestions about trade-off, right? So that you reproduce, but if you develop, more, then you don't reproduce. I know, but highly speculative. But it's interesting in that system, because if you think about traditional life history approach, high elevation, shorter breeding season, because it's a much harsher environment. They don't have a chance to produce more than once. At low, they have a chance to reproduce more. So what does it mean? It also means that you have to invest more into current reproduction, ideally, right? According to the life history theory. Because you only have one chance, they don't live very long, so you better invest as much as you can, and at low elevation, you may not. So we can test this by introducing these models of a predator for adult, like a hawk, a sharp-shinned hawk to them, and a control, and these birds are afraid of them, so basically if they will wait longer to enter the nest in front of a hawk, that suggests that they are investing less. They're taking less risks in order to compensate their chicks, right? So because if they delay it before going to incubation, that's my graduate student who did this work, then that's what it suggests, right? And so the prediction then would be that if the traditional life history approach works, then we should think that they should invest more. They should be much quicker at high elevation, enter the nest, and really ignore the predator, because it's very important for them to succeed at breeding, right? So again, that's kind of the basic idea, right? Long-lived, less fecund, self-investment, short-lived, more fecund, invest in current offspring, right? And so, based on this study, we thought the alternative idea may be that if we are dealing or we are witnessing selection cognition, and if the parental investment is another cost, then maybe trade-off. Actually, it would be the opposite result, that they would actually invest less, even though they have a shorter period, right? Maybe because they survive longer at the same time. Right, so here's basically the logic, right? Shorter breeding season, high elevations, few opportunities invest more in current offspring, but at the same time they have that better memory. And so life history predicts that all the traits connected to parental investment, clutch size, parental feeding, risk-taking should go in the same direction. However, it's a trade-off. We could expect to see some traits, some patterns, some traits to go up, and the other traits going exactly in the opposite direction. And so here's actually the view from the top of our location site. I think that was taken in June. And now we wish for the June to be like this, but because we get less and less snow. But usually we have, one of the years we actually have snow here till mid-July. And birds were breeding into the even late July. But so that's kind of where we're testing this type of things, right? So we have nest boxes, so these birds breed in them. We have hundreds of nests every year. So we looked specifically at when they start laying. We tested this during incubation and during fledging. The idea would be that if the birds delay their coming back to incubate, there's a cost, because basically temperature values will drop lower, and obviously there's a cost as well to delay feeding of their young, right? And so here's our models. The pictures don't do justice. They're actually quite good, right? And so we tested them. We basically used a randomized design where we randomly introduced an either way control, which was a dove or a hawk, to see if they'll respond differently to the hawk versus to the dove, right? So before, again, here's the breeding phonology. I want to show you the breeding time at high elevation is 2 to 3 weeks later. That's been pretty consistent, pretty stable. So there's a big difference in when they start breeding. So this is just, it's a Julian date, but you can see that the date for the first egg at low elevation, high elevation. Very consistent year after year. There's a pretty big difference, very little overlap between high and low elevation when they start breeding. So what we found is basically support for the trade-off idea, right? That high elevation birds actually were more hesitant to go to the nest at both stages when there was a hawk moth compared to low elevation birds. What's interesting at the same time, that high elevation birds laid larger clutches, significant larger clutches. So what we have is more investment in clutch, but less risk taking at the same time, which seems like a waste, right? So if you're investing so much in the clutch, why not to be, to do more with this, right? So we can look at again, so what we see is, again, big difference in clutch size, but when we look at the brood size, we actually see no differences. And the fledgling success, we see the opposite difference, actually, that high elevation birds have much lower fledgling success, and we have much greater nest failure at high elevation. And one of this year, when we tested this, it was 38% versus 14%. So that seemed to follow that graded study, suggesting, yes, they have better memory, they have better problem solving, they're actually laying larger clutches, which could be explained by their ability to secure better resources, but they're not delivering with these clutch sizes. So it's a big question why. At least this experiment shed some light that they may be much more hesitant, meaning that they're investing more in their own survival than they're investing into their own offspring. The question why, again, we think it's consistent with that idea. Obviously, we cannot say with this data that the selection on cognition caused this to happen, but nonetheless, it's a very interesting finding that goes opposite to traditional life history approach, right? Because we have high elevation, low latency to return to the nest, larger clutch, no difference in brood size, greater nest failure, lower fledgling success. And that greatest study looked at problem solving, and they found at least this variable exactly the same. So that they were much more likely to abandon the nest if disturbed at any point, right? So these data are inconsistent with the traditional life history because we have opposite direction for different components of parental investment. And it's more consistent, at least, with this cognition parental investment trade-off. So we still don't know we're getting now data with this bird, but we don't know difference in survival. That would be very interesting because if high elevation birds indeed survive much better, that may explain that too. And it may be more consistent than with the life history. Because meaning they have only one season to reproduce, or I mean shorter season, but if they live on average twice as long, well, here's the way for them to compensate for those differences, right? So we also think there should be other differences that restrict movements between elevations. If we're thinking about this divergence, if you think about local adaptations, if you have a full gene flow, that wouldn't necessarily work with it, right? So the question is then, what could be such mechanism? And one way for us to think about it was social dominance. So chickadees are very social birds. They live in permanent social groups throughout the entire winter with a linear dominance hierarchy, which means the dominant bird is the best bird. It gets all the food, it gets all the favors, gets the best mate, and so that's well documented in the entire family parida in chickadees and in tits, so it's well known. So then the question is, if there's a difference, then it may potentially prevent them from crossing elevation, right? So we brought these birds into the lab and we pitted them, in pairs and all possible pairwise combinations to see what happens. And what we found is that the low elevation birds are dominant pretty much all the time. So they beat up on high elevation birds consistently. And it's not even kind of funny because they're very mean to them as well. And the smallest birds are the meanest. So the size did not matter. So it was completely independent of any other variable we could look at. Basically, low elevation birds are just those bad asses kind of things, so that they just don't let any high elevation birds to get in there, right? And so it's interesting. So what we have is now one correlated with fast exploration rates associated with higher dominance, which kind of goes against the behavioral syndrome ideas because they suggest the opposite pattern. But what we see here, at least on the population level, we see this type of association. Again, I'm not implying that one feeds into the other. It's just they both are there at the same time. We also wanted to see why are they winning. So what's happening? Are they just more aggressive? And we did a basic standard mirror test, right? So we put a mirror in the lab and saw it. And indeed, low elevation birds, so you can see here the latency to fly and be aggressive, the low latency meaning more aggression. Once you put the mirror, low elevation birds take no time. They go straight to that mirror and they're fighting it like there's no tomorrow. So high elevation birds are not as aggressive. That kind of feeds into this, it explains it. So low elevation birds, they are dominant, and it looks like they're dominant because they're just more aggressive, intently there. So they're fast explorers, they're not as smart, they're basically bullies, right? So we have these bullies, they're just going in there, moving around, finding anybody who's not like them, beating them up and expelling them somewhere else, right? But that definitely is there, and that is a serious mechanism for them. So which means that if high elevation bird were to move to low elevations during initial dispersal event, that's not going to fare well. it's most likely going to die, because if you're low in the social dominance hierarchy, your chances of dying over winter are much higher. Again, that's been documented very, very well. Not in particular our system, but it's been shown very well, right? So what we have then, that low elevation chickadees, if they will move to high and disperse to high elevation chickadees, they don't have their cognitive abilities to do really well at high elevation, so they're more likely to die. And low and high elevation birds are very smart, but they just can't, they're not aggressive enough. If they go down, they're not going to do well. And again, their superior abilities are not, they're just not needed there, right? And so that, at least one mechanism that definitely should prevent a lot of mixing. It's not to say there'll be no mixing at all, but at this point, we just need some prevention for that to occur that will already will be maintaining if there's some adaptive processes occurring at this population level, right? So the next question we wanted to ask, are there other mechanisms that are there, right? So we wanted to look at songs. So here's Carrie Branch, my student who wanted to look at this, right? And so we wanted to see what's happening with the song at different elevations. And of course, when we think about the song, there are multiple competing hypotheses that may explain differences, right? So obviously, in our case, our interest was, is there a difference between elevations and high and low elevation? But one can say that difference can occur regardless of anything else, simply if singing at high elevation requires different output, because the air is not as dense as there may be different habitat. So there could be, in other words, acoustic adaptation would suggest that birds will adjust to the prevailing environment, right? Or it could signal local adaptation. If song is signaling to the female potential mates, then they need to know. To mate with local is important. If female is at high elevation, it doesn't want to make a mistake. It's going to have much higher fitness if it mates with high elevation male. And so the question then is, if there's a difference, it may signal that adaptation. It's well known in song dialects literature that females use different dialects in choosing the local males, right? And finally, we can also have temporal variation. If these birds don't move at all, if now we're talking about with genetic drift, meaning there's no genetic, gene exchange at all, then song can just drift, right? And then we can see these differences that are not meaningful potentially because it simply drifts because of no exchanges, right? So again, we have this very rapidly predictable changing environment in the mountains. So what's important about this, including chickadees, it's locally learned. It means the bird learns the song before it disperses. So it will sing the dialect that it learns at its hatch site. So no matter where you get it, it will still sing the dialect, right? And so we're dealing also with local male preference, or at least the idea of this, right? So there's several, again, like I told you, that there's several ideas of this. So we can look at acoustic adaptation hypothesis, when environment affects signal transmission, and song evolves to optimize this transmission. We have temporal variation, no movements, and song drifts, and we have signal local adaptation. So we actually compare two different mountains, right? Because you cannot do it on one mountain by default to kind of tease it, even to start teasing it apart. So we went to Sagehand and we went to Mount Rose, which is about 35 kilometers. Very similar patterns, right? So we look at the same elevation, and if you look at habitat, composition of pine forests, it's very similar, right? So what does it mean? It means that, for example, if acoustic adaptation hypothesis is correct, and there's a difference, then high elevation on one mountain should sing very similar song to the high of the other mountain, and low should sing very similar song to low, but there should be a difference between high and low and high and low, right? If we see that pattern, that would suggest that they're simply adjusting to high elevations, and it has nothing to do with anything else, right? So when we look at temporal variation, again, now we're talking about drift, however unlikely that may be, then we may see all kind of possible variation where things could be different entirely between this and this and that, right? So we can see high may be equal low and high equal low there as well, right? And we can see them being very different if they're just simply drifting apart, right? Again, that's unlikely, and actually we know that could not be true because we do have genomics data now, and we know we don't have genetic population structure in our system, which is definitely also rejecting that idea outright, that's definitely not happening. And again, if we are dealing with a single local adaptation, then we actually should see that all different should be pretty much unique, because we have different mountains. These populations do not come necessarily with contact across the mountain, so they're dealing, that's the boundary where they're crossing, then we would see the difference across the board in all four populations. So here's a probably I'll know the chickadee song, but I'll play it. Alright, so here's a pretty much standard chickadee song. So Kara recorded this. She measured all of these variables, and don't pay attention at this point, just all numbers. Mainly, it's a gigantic table. What you should see is that the difference are indeed different between the mountains. Some difference between high and low on one mountain are not the same. Like you see, these three variables are different at one mountain, not different at all at second mountain. This difference is different at that mountain, not at this mountain. So the differences are all over the place. And again, this is the same graph, so you can see a different, like for example, frequency ratio or start frequency. They vary differently. In other words, on one mountain we see difference in one trait, but we don't see it in the other mountain. But we see difference in the other mountain, we don't see in the first mountain. So when we plot it in a discriminant function analysis, we see nice 4 layers, which means all 4 mountains show different dialects, significantly different dialects. They sing different things, right? So that does suggest, at least or supports this local acoustic adaptation hypothesis, suggesting they may be signaling to their mates that they are better adjusted to this environment. It rejects the acoustic adaptation hypothesis. So temporal ratio requires no gene flow. We know that's not true. So this still is, so we were supporting this one, right? So we also want to see this climate variation. So what she's doing collecting now, because like I was telling you, we have pretty abrupt change at about 7,000 feet. So we're interested in to see that song drifts along elevation, like along the continuum, or it changes to a very narrow zone, or that snow zone where above 7,000 feet it's always snows and below it's always rains. And so we will know this by the end of the summer, right? So the question then is, and you can see also, I just a few pictures right of these guys, so you can just see that they are different. So this is one mountain and this other mountain, so if you've seen the sonograms. Well, does it matter? Do females pay attention, right? So we did the female preference experiment. So we had a female in this double cage, and then there's a divider, and there's a male from either the same elevation or opposite elevation. And we want to see if she's going to spend more time with the same or opposite. What we found is that females from high elevation are indeed highly selective. They like males from high elevation. Low elevation females had no preference. They really didn't care. So, interesting, right? It kind of makes sense again because these are under much higher selection pressure, at least hypothetically. So it's much more important for them not to make mistakes. Well, for these guys, it may not be as important. Of course, the males can make sure that there's no high elevation males there anyway. So it's interesting, but it feeds into this. So the results I'm not going to present here is that we also did a playback experiment by playing song from high and low elevation to high and low elevation males. What we found is kind of really puzzling, but we found that low elevation males are very aggressive toward high elevation males, even though they're dominant. High elevation males don't care. There's no difference in response. So we have high elevation females are being very choosy, but low elevation males are being exceedingly more aggressive toward chickadees from opposite elevation. Interesting. We can't still quite figure out why, but one thing for sure at least, they clearly can discriminate this one. They know. In other words, when they see a bird or hear the bird, they know whether it's from the same elevation or from a different elevation. There's no question about that. And that's kind of interesting, right? So in other words, kind of summarizing it, we have this large difference in spatial memory, we have no difference in development, or at least detectable difference, we have big difference in dominance, big difference in song structure, in male and female mate choice. So that all seems to jive with the general idea that we are dealing with some separation, where animals are paying attention and kind of may not prevent entirely gene flow. Of course, we are talking about just 7 miles. I mean, that's, and these birds can usually fly the distance within a minute. And yet, these things are pretty interesting. So they're consistent with the idea of natural selection. Of course, they don't prove it by any means now. So what we're doing now is we're actually trying to study specifically individual cognition in the field. So I just got a pretty nice NSF grant finally. And what we're doing is we've designed this spatial array. So as you can see, these birds are banded with RFID. So the feeder identifies the individual bird, and it either provides a reward, like you can see here, so the bird gets food and flies out. You know, this is another one, and then we'll see another bird that comes and it will not provide food to it. So hopefully this, nope, it's still providing food for this guy. But again, the idea on this one, yep, here it is. This one's not going to get much. So the idea would be is that you have basically a spatial, basically a radial maze in the field. They cannot learn socially because we train them first that this is antenna. It identifies, it reads the tag, and then it either opens it or not. Different birds is assigned different feeder. So you have multiple birds. They are assigned different, one of those uniquely identified feeders, and then we're trying to see how fast they can learn, because what you want to see is that the birds initially come, they'll go all over the place, and then they'll say, okay, only that feeder gives me food, so we can actually measure learning curve. We can measure curve, we can measure retention, we can measure the whole thing. So this last year we were able to tag 200 birds, and we've actually got measurements on more than 100 birds. And it's another issue that I kind of come more from behavioral ecology type background, dealing with small data sets. Now we have 120,000 data points that we internalize. So now we are dealing with a big data. It's interesting, they're clearly learning, so we're still now in the process of this. The idea would be that if we we definitely should be able to identify individual variation, then we can trace it to survival, we can trace it to mating success, fledging success, development rates, ex-pair matings, who mates with whom. We have more than 200 nests every year. So we really hope to be able to measure selection strength at different elevations and to connect all this individual variation in the field with the fitness. And that hopefully will provide us more data. Again, there are other outcomes looking at the genomics kind of things, even though I'm still not sure whether we can do it. We can compare genomes of birds that survived with birds that didn't survive because we're collecting blood samples from everybody that we're getting. Cognition is a complex trait, so we've done some genetics, we've done transcriptomics. We've been able to detect a lot of difference in a lot of genes that show differential gene expressions. But still, we're talking with cognition when we are dealing with probably multiple thousands of genes controlling this, and so it's a pretty difficult kind of task when we're and also we probably are dealing with a lot of other phenotypic differences, obviously not just cognition. I just showed you difference in so many other traits as well, right? And so the question again is, what's the key player here? Is the selection acting in cognition, everything else is falling with suite, or there's multiple type of selection acting on multiple traits at the same time, enhancing the survival. So in this kind of a pretty much on a wrap up. So this is a support that I've got over the years. NIH pretty much now stopped supporting international studies. They only care about humans, but NSF is still helping us. And here's my wonderful lab. And this is kind of what we have to deal with now. This feed is we're actually soldering all the computer boards ourselves, and we have to go and check them every other day in the winter when it rains or snows. And if you've not soldered or dealt with computer exposed parts in the rain or snow, it's a very kind of fun business. So I highly recommend after driving the snowmobile for 2 hours and having your hands freezing and everything, and then you have to deal with little wires, and they always, things always go bad. When people say everything works, no, it doesn't. All right, and with this, thank you very much. Investments. In our case, now we provide some sunflower seeds as well to them, but they are specialists on pine seeds. They're very dependent. They are caching tens of thousands. The other interesting thing is because, of course, pine success varies, and so they should be able to fall on bad years. We have not seen this yet because of this weather changes. We actually had amazing pine seed production 2 years in a row, which was just shocking to me. And these birds take pine seeds over feeder, so our work slows down when there's a pine harvest, because then they don't want sunflower seeds. Which you wouldn't do, right? If I give you pine seed and a sunflower seed, you'll go on pine nuts. That's smart.
So, sorry. Okay, I'll go. So given you've got all these birds banded, and have you done any extra pair paternity work?
Not yet. We want to do it. Again, we just banded this last fall, so we just started. I just got that funding, and so we just started massive. We're going to continue doing as much as we can, so I expect that we'll be banding 300, 400, 500 birds every year. So we should have a lot of, because we will identify them by age next year, so we can actually estimate survival also based on age, because juveniles supposed to die earlier or not. So we should have all of this data, yes, and we will be looking at ex-pair paternity.
So you touched on this really briefly, Vladimir. I want to confirm you don't find any population genetic structure differences.
No.
Okay.
Unfortunately, I really wanted to find it, and it's not there.
But in some ways, it's actually a more interesting puzzle that there's not, and yet you're finding.
I agree. It's kind of interesting because now, kind of historically, when we're publishing things, we've been every time we'd mention that we think there's selection, we had reviewers saying, remove the word selection from your entire manuscript and never mention it again, ever. Mm-hmm. And we say, why not? And they say, unless you show us that there's a genetic difference, you cannot talk about selection. And we said, no, actually, if you open an evolution textbook, it will tell you that local adaptations, genetic adaptation can occur despite gene flow. Lots of works on birds, and particularly teals in Andes, for example, showed this beautifully. Even in Darwin finches, they showed speciation with gene flow, right? But that's kind of I really wanted to find it just because to say, finally, here it is, you want it, here it is, but we didn't find it, so now we're going to say, no, it's not there. But we actually don't need it. No, I agree with you. I think it's even more interesting.
Yeah.
The question is, what's the strength of selection that we're dealing with, and whether that can maintain and generate these things. And now, if we're monitoring it over years with this crazy fluctuation in the climate, how that's going to affect it, because one can expect if the winter is mild, survival is going to be up.
Yeah.
And so the difference in cognitive abilities may not matter as much. The good thing that even in the winter like this, as bad, high elevations are pretty tough. I mean, yes.
Do you think you could cross a high and low elevation bird and release it? They won't.
Well, they will go straight back to where we are. We can try to do it in the lab, but breeding in the lab is very tough. Even because we were thinking about cross-fostering, but differences in reproduction makes it very hard. Because basically we'd have to take the latest nests in one and cross-foster them to the earliest in the other one, and that's not random. That creates a problem, so we can't do it.
So do you think you're, so one of the things we struggle with some of our work is trying to tease out the timing of reproduction and the role that plays relative to aggression, and that they sort of confound each other because, you know, often our low elevations are starting with a 4.
The low elevations are starting like 3 weeks early.
And so you could imagine in that situation that the aggression is different as a consequence of, you know, you've already bred. This is a chance to get another opportunity.
Potentially, but we see this aggression like in January and February, so
it's
yeah, I don't think it's connected to that. Yeah, I think breeding is just simple, just climate thing. Because like right now, a low elevation, I know even Sage-Can, it's summer.
Yeah.
It smells like summer. The dust is already there. The birds are singing, and you go to high elevation, and it still is frozen. I mean, it's not frozen like Alaska frozen, but it's quiet. Nobody's singing. The birds are still going like crazy to the feeders. I mean, there's a lot of other things. I mean, there's a lot of other difference, which is interesting. Even coming to the feeders, the birds at high elevation, they're just fierce. I mean, they just go like there's no. They just go like life depends on it, which is kind of very interesting. And even though we found no difference in neophobia, that doesn't necessarily like when we're there, the high elevation birds literally almost land on us. When we move all the things, taking memory cards, low elevation birds, they stay far. So suggesting that, yeah, I mean, it looks like it's tough at high elevation. The question is, do these birds have it easy with its better memories, or they still they suffer even higher mortality? And we should have the answer to this.
Any other questions? Well, thank you very much for a wonderful talk.